Let's welcome Dr.
Thank you. Thank you. So I thought I'd just tell a little bit more about on the open side of the SGC model, because Nicola already introduced to many interesting molecular biology techniques, but what else are we doing? And secondly, why are we doing the open? Yes? But as with everybody else, I thought I should start with a little biography or a little bit of what I did or what I'm doing. As you can see, I normally don't use my characters in my presentations. But as you can see by my name, I was made in Taiwan in an age when they were not yet doing great stuff yet. But then my family emigrated, and I moved to a very different country just to get people awake. 3 chances, come on, just get a guess. Any country. It was mentioned today already a couple of times. Okay, no, sorry, strike one. Brazil, there we go. Well, there we go. So that's very fast. Exactly. Well, I hope I didn't have it. Yes, I grew up in Brazil. My parents moved there, and it was a very, very lucky existence. So I had a great time there and decided to study biology. And why did I decide to study biology? Well, unfortunately, I have a brother who has autism. And as we know, there's no treatment for autism. And in fact, the term autism is a very big term. Anything that doesn't fit in certain diagnostics, that has any problem with social interaction, put it in the bag, right? And think about this. Many of the diseases we know today still are based on this very old conception. We talk about cancer. Who knows where the word cancer comes from? Where else can you see the word cancer? Every day in newspaper. Horoscope. What is the sign? What is the sign of cancer? It's a crab. Why was that? People start opening. People were dying, and they saw this mass with lots of ramifications. That's how the name came about. We're still calling that disease a crab. Whereas in reality, now we know, cancer is not one disease. Cancer are thousands of different diseases with different etiologies. Why are we still keeping calling that? Right? So, that took me always to think, oh, why can we fix this? Why can we cure this? And of course, as my Chinese mother, she would always push me either to medical school or law. So I said, oh yes, how many people can I really treat if I was a medical doctor? Quick pen and paper, how many days per week I have to work, how many hours, X number. However, inspired by many scientists, I said, wow, scientists actually do a lot of great good that are much more durable in time. So why don't I start doing this? I was lucky enough to get into a great university in Brazil and did my degree in biology, and I moved to a master's in human genetics, clinical genetics. And then I was always impatient. I just couldn't wait. DNA was coming on vague then. So okay, what's after DNA? Proteins. And luckily enough for me, at the backyard of my university, we had a very good equipment, synchrotron. So again, there's no many universities that have them. For many years, Brazil was the only country in the entire southern hemisphere to have a synchrotron. And I was lucky because that's at the backyard of my university. So I started working there. Instead of actually, guess what, learn how to do crystallography, my supervisor, who was a Russian, didn't speak Portuguese, showed me a Unix computer and say, here's the computer, here's the data set, here's the manual, here's a pair of spanners, because we haven't built a beamline, good luck. So that actually got me into the thing. Why? Even if you started biology or pharmacology, don't get restricted to what you are boxed in. That's a stepping stone. You see from Hagen's beautiful experience that although the start was chemistry, now Hagen's everywhere, isn't it? Really, really going out there, solving problems. And that should be how you should see your future careers. Pick a problem that keeps you awake at night every day and go for it. After my PhD, I went to spent a couple of years in San Diego at Scripps because I couldn't do crystallography anymore. It was taking me 3-4 years to solve one protein structure. Today it's still around the same timeline, but then I'll come to the SGC later on. And then I went to do the computational side. Can I just do simulations? Much faster. Then family, my wife then, my wife now, my girlfriend then said, oh, you picked the first postdoc. we were here in San Diego. I want to move to France. So we moved to Paris, spent a couple of years, and then the SGC started in 2004. And I said, well, Oxford is a good place. I came here for interview, it was 38 degrees, entire week. I went back to France with a tan. They said, oh, I thought you were in roast beef land. What happened? No, I was there, but it was sunny. I moved here and couldn't leave. The SGC started with this completely open science platform. And what do we mean by open science? It is really thinking about open source, somewhat like Linux software applied to medical discovery. Okay? Now, this is what keeps me awake at night. Does anybody know what kind of disease is this?
I know in Korean, but not in Korea.
Ah, right. How do you call it in Korea? So how do you translate that to? Ah, so if you look carefully, you do have a distortion, but look here. Are you expecting to have bone in this area of your body? Do you expect to have bone here? Do you expect to have bone when you normally see muscle? No, you don't. This is
carry on.
this is actually an ultra-rare disease called fibrodysplasia ossificans progressiva, or the stone man syndrome. What happens is that the muscle slowly turns into bone. Connective tissue turns into bone. Kids have terrible existence. They come to a stage in their lives where they have to decide whether they want their jaws locked closed or open. Okay, they have to make that decision for their life. They have to make a decision how, what's the position they want to be locked in, because it's irreversible. It's ultra rare. It's 1 in 2 million people have this disease. What happens with this? Do you think any pharmaceutical company would develop any drug for this? 1 in 2 million. In the entire UK, you won't have more than 30, 40 patients. So it's a big failure there. Nobody will look for these kids. And yet, they exist. Imagine being the parent of such a kid, and they come to us. And they ask us, what can you do? I'll come back to this story later on. But that really what keeps awake at night. And what really bugs me, Nicola, and all of our colleagues at the SGC, is that innovation in biomedicine is really, really risk averse. You go to any company, they're working all on the same targets, trying to do the same drugs. This is actually an editorial that came out in Nature that one of our colleagues wrote. And this exemplifies clearly what's happening. There's so many routes, so many ways to attack a problem, and yet everybody takes the same route because they don't dare to risk. But don't worry, we have academia. Academics are very smart. We'll save the day, right? Well, let's see. Who knows about kinases? Class of protein. Great drug targets, right? Control many of the cell signaling, cell cycle, and perhaps they're involved in that disease, terrible disease that I showed in the beginning. But they are actually involved also in many different cancers. So you cannot say that it's a small family. You cannot say that it's a family that has no big, if you like, potential to be a drug. And yet, if you take PubMed, everybody will use PubMed, there are 520 kinases in the human genome. Type each one and count how many papers comes out. And this is a scary distribution. More than 90% of all the work ever done on all the 520 kinases revolves around 50. Even though you have very good evidence, RNAi, driver mutation, and so on, to show that many of these are the ones that are really relevant. Why are we not working on this, right? P38, the kinase domain of P38. There are more than 15,000 papers on P38 kinase domain. There's probably more paper than the number of atoms in the kinase domain. Have you ever stopped to think about that? It is stupid. Why? Now, you go to your PhD, the first thing your supervisor will say, let's pick a project. Let's pick a safe one. Which one? Oh, P38 is a good one. Oh, you want to do the kinase 351? You go to your funding agency with a paper, piece of paper A4, back and forth, saying, there's one paper saying that 351 is important, and I want to know why. What do you think will happen? Unfortunately, the funders are thinking more like industrial funders. What is my return on investment? Instead of actually saying, we need to explore the unknown. Okay? Oh, and now you know what is worse? Oh, perhaps pharma industry is smarter. Let's see the patents filed on the kinases, okay? Guess what? Because it follows. Pharma still also saying, well, the academics are smart, let's follow what they do. We all read the same papers. We all came with the same ideas. Smarter scientists in Korea will put a grant to the Korean funding agency. Some smart people in the US will put it on the US funding agency. Some people in France will put it through INSERM. Some people in Brazil will put it through FAPESP. What we have now, hundreds of projects that are very similar because they read the same paper, and only one will win the publication. All the rest fizzles out. It's stupid. Even with so many diseases and so many different opportunities to tackle on. And by the way, you know that disease that I show you, the Stoneman syndrome? There is one mutation, change 6 atoms on a kinase domain on this kinase, for which very little has been done. But I'll come back to that later. Now, another problem is that people think that great idea needs to be protected, patented. Yes, when you can figure out the value of that, that's fine. But a lot of people put patent way before they know what they do, what gene does. And when they do that, instead of sharing information, they start locking it back. They start protecting without sharing. Biology is so complex. It is easy to fix a computer because we designed this piece of machine. We know the blueprint. It is impossible at the moment to fix everything in the human body because we didn't design the system. It's very complex. Why are we still segmenting small 20 people's group to tackle really large problems like Alzheimer's? Great example is TRPV1. That is one calcium ion, calcium transporter is a membrane protein, back again membrane protein. Was discovered in the early 90s, late 90s, 97. That's a receptor for capsaicin, so the chili pepper, hot. But also is involved in pain. And some people said, well, perhaps if we create an antagonist, an inhibitor for this channel, we might shut down pain signal. It would be great drug medicine. What happened is that, and then a start race, so everybody started filing patents on TRPV1. We don't know whether it's possible, but let's file a patent. It's very small here. I normally use very big projection. I will read what are the indications that people are putting patents for TRPV1 per year, okay? So 2002, 2003, 2004, and this is a number of patents related to what kind of indication, what disease. So this purple here is pain. I'll just move over and read it for you. So the first one is pain. Second one is neuropathic pain, or still pain, that's fine. And then we start. Overactive bladder, incontinence, allergy, Alzheimer's, anxiety, arthritis, asthma, cancer, COPD, depression, diabetes, fibromyalgia, and we go on. What does it look like? Wild West, snake oil. Oh yeah, this will cure everything. We stop here, but there are patents that claim this will cure everything. Why? Because this is just a matter of bet. It's not based on real science. How do I know? You go to literature again, PubMed, type TRPV1 and many of its synonyms. How many papers are here in 2005? 167 papers. How many patents in 2005 for TRPV1? More than 200 patents. Where does that come from? And, make things worse, finally in 2005, somebody published the paper. Pharma were working on this early. And they found out that if you inhibit this calcium channel, you block the pain, but you also cut off heat sensing in the body, temperature monitoring. So what happens is that patients that have, in models that you have TRPV1 inhibited, you have hyperthemia. So yeah, you won't have pain, but you burn to death. Is it a good drug? Perhaps not. And yet, after the paper was published, people keep on patenting. It is crazy. And now you wonder, why 9 out of 10 projects in pharma, they fail in phase 2, which is efficacy. Because all this is a matter of shooting crap. It's actually playing a casino. You put a patent, you say, I claim this as my idea, without even any fundament. And when you go down to the time to really prove which disease indication you have to find for your compound, you will fail because you don't even know what it's there for. How can we revert that? Stop that. Stop complaining. Think about why should we compete in an area where we know so little? Why don't we create a zone, a public-private partnership, where everybody, all the pharma partners, all the governments, because they do have patients, they have population to treat, all the academics, put the resource and money and knowledge into one common place and say, with this amount of money, we'll do one, everything open, so we all learn with the results. Two, everything new. We will not work on those 50 kinases, we work on the rest, because we just want to raise the knowledge, right? And then three, let's make it completely patent-free. Whatever we discover here, no one owns. Everybody can use without even have to ask for permission. Push it away. It sounds like a pipe dream, doesn't it? But what we promise to make available are structures of the protein, so 3D, all the atoms mapped on the protein. So you can look at the active site, for instance, design a chemical compound, a tool to inhibit it. Antibodies, as research antibodies, screening and cell assays, everything put out in public domain. Once you figure out what that protein is doing in different diseases and different systems, then the race can start. There's still a lot of money to be made down here. We don't care. What we want is more shots on the go. Yeah? Any questions so far? Please interrupt me. We love to chat instead of actually one way, okay? Now, we hate duplication, so I will not talk about this slide. So I'll give you an example. We heard a lot about genetics. Who has heard about epigenetics? Epigenetics, of course, right? So you can think that the correlation between genetics and epigenetics is the following. Imagine a recipe book with all the best recipes from Korean culinary. You have street food to very fancy food, right? It's all in there, the genome. But if you're throwing a 3-year-old party for Nicola's baby, what kind of recipes would you use? You use different sets, right? From the same recipe book, you can extract different parts of information. There's another layer of control on the DNA, which is controlled by this epigenetics layer. One way could be tagging the DNA with methyl groups. Another one is the smart way that nature is actually now structuring or organizing the long piece of DNA that we have into nucleosomes. Remember the nucleosomes? DNA, histones around, DNA, another blob of histones, turn around. Yeah? So they form different volumes. The beauty of that, each one of the histones has a long protein tail, and each one of these positions can be modified with MET2 group, AC2 group, other groups. Think of that as barcoding. You have a really specific barcode for each one of those nucleosomes. Now, a lot of drugs, now there are no new generation of drugs for cancer called HDAC inhibitors, for instance, that actually affects how enzymes add those barcodes. But if you inhibit that one, you're messing up with the writer. It's very, very bad. What you want to do is inhibit the protein to then read and understand those barcodes. These are the epigenetics regulators, the readers. What we talked about, it seems to be a good way of working because that's a completely new layer of regulation. When we talked about our ideas to many of our pharma partners and colleagues, they said, forget it won't work. It's protein-protein interaction, there's no pocket, it's very shallow, forget it, it's very slim chance. We said, no, I don't think so, because we've solved structures. We solved structures, there are 61 of these in the human genome. We solved about 35, 40, and say, well, look, there are actually well-defined pocket clefts that can read the lysine modified with dissected group, for instance. And we know the surface properties, electrostatics, for instance, so we can design a very specific compound. So GSK said, okay, that's fine. Look, there's an old Japanese paper. Have a look. We look at some chemistry there. And in 6 months, we designed a compound specific for one of those genes, BRD4. What happens then, when you have a specific key compound for a completely novel protein, you don't know what the protein is doing. What you do? You look for a disease where this protein is involved. And by chance, this protein is involved in a very rare cancer called nut midline carcinoma. Normally diagnosed in kids. And whenever they have found the disease, they have 6 months to live. The very good doctor in Boston who actually had access to patients and had actually extracted those tumor cells and put in rats so they can grow. So we said, hey, Jay, why don't we work with this? Jay Bradman is his name. So we sent a compound, and Jay said, oh, yeah, sure, why not? This is a colleague of mine, Panais, who's a fellow in Balliol, by the way. And as Panais called Jay and said, hey, we can send you. And Jay said, oh, great. Should we get the lawyers in? Because there must be some transfer or license or patent. We said, no, it's free, there's no IP. Hang up the phone, FedEx the thing, start the work. From the phone call to doing the experiment, writing the paper, sending it to Nature, and publishing, 11 months. Typically, if you're dealing with universities, tech transfer office to say, can I use that or not, you will probably be still thinking about, can we start this project or not? It's still on word of words. For the first time, it was shown that these bromodomains can be inhibited using a compound. And we don't hide what the compound is, we show what the compound is. And better, we said, anybody who wants some milligrams of the powder, write us, we send it to you. The same thing we used, you can use in whatever you like. You can try to reproduce our experiments, we're not afraid. It is that good. A month later, more than 100 labs asked for the compound, we shared. And what happens is that the best experts in every single disease that had access to good disease models start testing. Oh my goodness, I didn't know that if I inhibited BRD4, I had a reduction in the count of leukemia cells or myeloma cells. Who would have thought? It's almost reverse pharmacology. Instead of saying a target is involved in disease, we say our target is important, let's find the disease. So much data that then people start publishing a lot. There's so much data that in less than 3 years, since we started pipetting, there's enough data for a pharma to say, thank you, we've seen enough data, we're going to launch this into clinical trial. That normally takes between 6 and 30 years, depending on the projects, because it's very slow to accumulate a project information all by yourself, alone. It's a crowdsourcing. And of course, now, this one probe has already enabled, the paper was published in December 2010. Today, there are more than 40 clinical trials registered for many different diseases by 10 different companies with 10, 12 different compounds based on this, but targeting the same protein. That alone has already means that the world has put in more than $1.2 billion of investment in something that nobody believed here. All right? So that really changed the spectrum. You don't live here, but recently GSK, one of the largest UK pharmaceutical companies, they cut out 150 programs. The few they kept was this one. Right? And that brings me to the final part of the talk, which is we love doing science. But what we really love doing is actually expanding the limits of science. Science is not just science, it's not just knowledge, it's a betterment of society. And that means that by making it open, we can work not only with academics, we're open, but we can work with non-traditional partners and stakeholders, because they can actually help us tell which is right, which is wrong. Yes? We are scientists, we look things in a different way. But ask the patient whether they can wait for an extra year, because we're just discussing whether the reagent can be traded or not. You can't. So this is a program we designed. We say I'm a patient because every single one of us will be patient one day. Every single one. We will hit the disease for which there's no cure. Eventually. Right? Alzheimer's is just around the corner for people like me. I'm getting old. But the more important thing is that what the patients need is a medicine, and what they don't have is time. So we create a program, what we call Impatient. That's what we really are. We are really impatient. By giving access to our huge platform of drug discovery. To who? To small, rare disease groups. Because they are so small, they collect money in coins. And what they can do? We're talking about multi-billion-dollar companies. And they can collect £30,000 per year, £80,000 per year. How do you do that? Well, by working with open access models. I'm coming back to the first slide. FOP, the gene was discovered in 2006. Sequencing, great. But what you do with that information is sitting on the shelves. Because nobody wants to work on this. You cannot write grants on it, farmers won't pay for it, it's dying. They came to us, to our colleague Alex Bullock, and Alex said, sure, I will solve the structure for you, solve the structure. Because we can solve the structure using proteins, for instance, that Nicola produced, we can actually test the normal protein, see what's the activity of that kinase, and then produce the mutant protein and see what's the activity of that kinase. Luckily enough, we saw that the mutation caused a gain of function. That means that the kinase was much more active, which means that if that happens, we can design a compound that can block, right? So we found about the mechanism, the molecular mechanism, and in 2012-13, we started working with academics in Harvard who also believed in the model to generate new compounds. And then we tested in our assays. There's another group who also made accessible their animal models. So we put this the animal model of the disease. You can see that bone is forming here on the inner thigh of the rat. But when treated with a compound, you don't have that anymore. Right? And there's so much going on that Alex, he said, let me have a look at some of the existing drugs that were not approved for their indication. They were testing different cancers. And found out that a compound from AstraZeneca called saracatinib, again, we're not afraid of saying, what can they do? Sue us? They can't. It's research. Saracatinib can inhibit this kinase. And now, from something that when you have a diagnostic means a very painful and slow deterioration and death. I mean, these kids, when they get cold, even the inner lining of the lung gets turned into bone. Right? It is very, very painful. And you can think, oh, why don't you operate? Because every insult you cut, that means that because you cut, there's a wound that will be turned into bone as well. Right? So you cannot operate this. In 6 years now, Alex is designing a clinical trial for the disease, for something that's ultra rare. And what's most beautiful than this is to know that this same gene, and that's another really good example, we don't understand biology, is involved in a rare brain cancer. Same mutation. How does it happen? We don't know. And when they found out about this brain cancer, they contact the brain cancer charity, contact us, say, we know you have reagents, can we get it? And Alex said, sure, send everything. So instead of actually spending 4 or 5 years doing this, they cut down 4 or 5 years. And now we're designing new compounds for this cancer. I won't show this video, it's that's what I'm saying. And that's really the last slide. So I think between you and me, between Nicola and I, we showed you that we do have a lot of research going on. We do work with disease that very few people will be working on, rare diseases, for instance, or parasitology and neglect diseases. We're going to very high-risk areas like neuroscience and psychiatry. We're working with patient disease foundations. And because the way we operate, we can really introduce really dramatic change the way society looks at drug discovery. So at the moment now, we have a program with the Oxford Martin Schools, a think tank here, just yesterday, and with the evidence for governments and policymakers to start incorporating this kind of model into future policies. We are in direct contact with the European community and with the UK government and with the Brazilian government, of course, which I'm very proud to be able to revert back and cycle everything, right? So with that, thank you very much for your time and attention. I guess you're all hungry and thirsty, so thank you.
Okay, any questions, please? Food is ready, but if you want to ask, I'm sure you are hungry for food, but maybe you can ask him questions. After eating, you may be hungry for questions. But any questions?
Is the clinical candidate drug is in clinical trial, or you just try that candidate in one person who has that disease?
Right. So here, you mean this one? Right. The clinical candidate already exists. It's a compound by AstraZeneca called saracatinib. It already failed phase 2 for the original intention, which is for cancer. It was designed for cancer. It failed the trial. So, AstraZeneca made it available. Whoever would like to screen it, who knows? They can find something else. So Alex found that that compound specifically was inhibiting ACVR1 very well. So we now have both the saracatinib, which is already in phase 2, so it's safe. AstraZeneca said yes, because it's only 30 patients in the UK, there's no money. They said you can take that and do your clinical trials, but we will not pay for any clinical trial. So Alex has now raised funding to fund a clinical trial. The challenge with this is there's so few patients. You cannot do a double blind. Ethically, it would be wrong. Yes? So you have to put everybody open label. You have to get everybody to get access to that. However, and that's the sad part of it, different organizations, they also like to claim they help the patients. So there are one or two other compounds out there for different pathways for the same disease. But they too are saying, these are my patients. Imagine 3 or 4 different clinical trials with 40 patients. Impossible, right? So something is not quite correct. That's why we want to bring economists, IP specialists, ethicists, and social scientists to work with us, especially on the rare diseases area, and come up with a good solution. So you cannot do this. It's not morally right. Yes? So yes, and of course, these 2 compounds we are now developing is going to preclinical development. So we have not given up on those 2 compounds yet that we've designed.
Just I comment that your talk was really great and is even touching. Very expert. And I have one quick question about the kinases. I thought, maybe I misunderstood so far that the kinases were searched for the drug targets so far. And over 500 the kinases, so only 50 or less than 100 is all focused, but the other 40, 100 something is unexplored.
Yes.
I was thinking that the kinase, the unexplored parts, are not much druggable, and then maybe just people are trying to look for other types of, rather than kinases like ubiquitin. But you showed beautifully that the one example which was not focused yet, but you showed all those clinical evidences. So for your opinion that all other, not all, but still that many of those kinases are still, it's good to try rather than you focus on other types.
Exactly. So about non-druggable, druggable, that would be a great argument if you're looking at, for instance, membrane proteins. Some of them are not very amenable. Some of the membrane proteins, actually, some ion channels, for instance, they actually is modulated also by the composition of lipids around it. How do you tackle that problem? Now, when you look at kinases, all the drugs that are being designed now, they hit the ATP binding pocket. And all kinases will have ATP binding pockets because that's their life. They take ATP, transfer to phosphate group. So in that sense, all of these are good targets. The question is whether it can be specific or not to tune in just one kinase or another. But there are lots of good evidence, and that's thanks to work of structural biologists, to say that some of the kinase inhibitors are now pushing conformational changes and are finding new pockets. So yeah, there's a big hope.
Thank you.
Okay, thank you very much.